You are looking at a real human eye. Now, the outermost layer of the eye is what's known as the fibrous layer, and it's made up of the white, accurately known as the sclera, as well as the cornea. You can see that fibrous tunic. There's the sclera, the tough outer layer. As it's connecting, it's continuous with the cornea. So this is the cornea that I'm poking with the probe. And then they're going to go around, and eventually in the back, we meet the optic nerve. So this is where all the neurons that are going to be heading towards the brain. Now, if we look at this red portion, is what's known as the vascular tunic, which is where all the blood supply goes. And then the internal tunic is this film. So if you look closely, you're going to see this white film. That's actually the internal tunic or the retina. That's the photoreceptive layer that all the rods and cones are going to be placed in. This structure here, this is the lens. This is just a crystalline network of proteins that will help further refract the light. And this right here is actually the iris. So if you look closely, you might be able to see the iris, which is actually two different muscles called the dilator and sphincter pupillae muscles. And then there's a hole in the center of them called the pupil. In this space, there would be a humor called the aqueous humor. It helps provide nutrients to that cornea, but light is going to go through that cornea, be refracted through the aqueous humor, go through the pupil, and then the light is going to go towards this lens. The lens will then refract it once again into this space. Now this space would normally be filled with another humor called the vitreous humor. And that light is then going to go towards the back of the eye to that photoreceptive retina. And again, there's one small area that we can't really see in this dissection, but it's called the fovea. It is where all of the cones, or at least the vast majority of your cones, are going to be located, and cones detect color. The rest of the retina are going to have rods in them, detect low light sensitivity, so they're really kind of like black and white vision. But all the cells inside of the retina are then going to be sending their signals that are then going to go through that optic nerve and then just make their way to the optic chiasm and all the way to the back of the brain.